Welcome to my tutorial. The learning objective for today is what is DNA and how does it work? So what does DNA stand for? Well, DNA stands for deoxyribonucleic acid. Now that's a pretty difficult word to remember and even more difficult to spell. Now a really handy way of remembering deoxyribonucleic acid is to do the following. See how many words you can make from deoxyribonucleic acid. E.g. riddle, it's a 6-letter word. You can make it using the letters in deoxyribonucleic acid. What's the longest word you can find? Try and find at least 15 words from deoxyribonucleic acid. And by constantly looking back at the word, you'll commit the spelling to memory and you'll remember it subconsciously. And then hopefully, if you're asked in the exam, you'll remember that it stands for deoxyribonucleic acid and you'll spell it correctly. So this is the sort of question you might get in your exam. Can you list the following structures in order of size, starting with the largest? Now, I've asked you here to start with the largest. An examiner might ask you to start with the smallest, but read the question carefully. Don't make a silly mistake in your exam. So what I would like you to do, I would like you to pause your video and try and put these structures in order of size, starting with the largest. Right, pause your video now. Right, okay. These are the answers hopefully that you would have got. So did you get A, D, B, C? With the biggest being the cell. And then you can see that within a eukaryotic cell, you have a nucleus. And that within the nucleus, you have these structures called chromosomes. And chromosomes are made of DNA. And what a gene is it's a short length of DNA that codes for a protein. So the gene would be the smallest structure here. So what actually is DNA? Well, DNA is your genetic material. We find it within the nucleus of a cell. We know it stands for deoxyribonucleic acid. But if we had to describe the structure of a DNA, that's slightly more complicated. So if we look at this diagram here, what do we have here? Well, this top diagram shows something called a nucleotide. And a nucleotide has got 3 components. It's got a phosphate, it's got a sugar called deoxyribose sugar. It's actually that deoxyribose sugar that gives its name to DNA, deoxyribo from deoxyribose, that sugar. And then it's got a base. Now there are 4 different bases that can be found within a DNA nucleotide. They're called adenine or A, thymine or T, cytosine, C, or guanine, G. So a nucleotide is made of a phosphate, a deoxyribose sugar, and a base, A, T, C, or G. Now it's these nucleotides that join together to form DNA. You can see this is a structure of DNA. And can you see there is a chain or polymer of these nucleotides going in that direction? And then can you see here there is another chain or polymer of these nucleotides going this direction? So can you see that DNA is actually made of 2 strands? You've got this one and you've got this one, which are polymers of nucleotides, hence polynucleotides. So it's made of 2 polynucleotide strands. And those strands are joined together by their bases. Now, these bases join in a very particular way. Base A will only have a base pair with a T, and C will only have a pair with a G. And what holds them together is these weak hydrogen bonds. Now, you can't make the bonds holding the 2 strands together too strong, because as you'll learn when we do a tutorial on replicating DNA or protein synthesis, you'll know that these bonds sometimes have to break. So DNA is made of nucleotides, which are joined together to make polynucleotides. You have two polynucleotides joined together by hydrogen bonds between bases. So you can see this gives a ladder shape. Now this ladder is actually twisted into a structure called a double helix. It's the order of the A's, T's, C's and G's, these four bases, which actually provides the genetic code. And it's your genetic code that determines the characteristics of a living organism. So, put simply, DNA is a polymer. It's made up of two strands or two polynucleotide strands, and they're coiled together in a shape of a double helix. I'll say that again just to try and reinforce that point. DNA is a polymer. It's made of two strands, two polynucleotide strands, joined together, and they're coiled in the shape of a double helix. And that double helix is that twisted ladder shape. Okay, now on this diagram you can see these are called the sugar phosphate backbone. And that's just because the uprights of the ladder, as we can see on this previous slide, were made of, you can see phosphate, sugar, phosphate, sugar, phosphate, sugar, again here, sugar, phosphate, hence why it's called a sugar phosphate backbone. Right, okay, let's move on then. So what are chromosomes? Well, a chromosome, this structure here, or these structures here you can see in the electron micrograph, taken using an electron microscope, these structures here are made of DNA. Now, in these particular diagrams, you can see chromosomes as X-shaped structures. They don't always look like this. Chromosomes are X-shaped after the DNA is replicated and just before a cell is about to divide. The DNA coils up and condenses, and you see these lovely, neat X-shaped structures. It's where the DNA is super coiled up. There's actually proteins in a chromosome as well, which the DNA actually coils around to give that shape. But GCSE, we usually just say that chromosomes are made of DNA. So in a human body cell, we would normally have 46 chromosomes, and 23 of those would come from your mother and 23 would come from your father. So we sometimes say that you have 23 pairs of chromosomes, which means we have 46 altogether. Now again, if you were to look at a chromosome, you can see that a chromosome is made of supercoiled DNA. And if you looked at the DNA in detail, you would see that you've got the base pairs in the middle. And if you read along the sequence of base pairs, in one direction on one strand, that's known as your genetic code. And it's that genetic code that is unique for all human beings. Well, except for identical twins, they have the same genetic code. So if we were to look at the chromosomes from a human body cell, they might look a bit like this. This is where they've been put neatly together, they've been matched, because you know that you have 23 pairs. And you can match those chromosomes according to their length. You can see where the centromeres are joining up the two strands. You can match the chromosome. Now it's this 23rd pair that you can see look very different here. And that's because this 23rd pair is the sex chromosomes. They determine the sex or the gender of the human being. And this organism here, from which these chromosomes have been taken, is a male. And that's because the 23rd pair are an X and a Y. The sex chromosomes for a male are different. For a female, they would have two large X's, but for a male, there is a large X and a small Y. So We know that a gene is a short section of DNA found in a chromosome. We know that a gene codes for a particular sequence of amino acids. We know that it determines which amino acids are joined together to make a specific protein. And the actual sequence of DNA, this genetic code, can actually be copied and passed on to future generations, to the offspring. And that's why we inherit characteristics from our parents. So, a quick checkpoint then. What is DNA and how does it work? So let's have a look at these diagrams. What do we have? Well, we have a length of DNA here. That's a gene, okay? Genes, lengths of DNA, will dictate which amino acids join together to make a protein. And proteins are required for building cells. Your cells join together to make tissues, and tissues join together to make different tissues join together to make organs. Different organs make different organ systems, and then the organ systems will make up an organism, like a human being here. Now, there are actually 20 naturally occurring amino acids, and what amino acids join together and how many of them join together is determined by the gene. So it's worth looking back at this diagram if you need to when you answer the following questions. So add the stages in the correct order. So what you've got is a diagram. What I would like you to do is I'd like you to read A, B, C, D, E, and F, and then use those labels to decipher the order going from this human being here right to this part of DNA molecule here. Right, pause your video now and have a go. Right, okay, hopefully you would have got the following order: C, B, D, F, E, and A. So what do we know? We know the human body contains about 100 trillion cells. That's amazing, isn't it? Considering the first thing that was ever used was a fertilized egg cell, a zygote, and that mitosis, nuclear division, has resulted in an adult human being being made of 100 trillion cells. Then, of course, you've got a nucleus inside each human cell, except for red blood cells, they don't have a nucleus in them. And then each nucleus of a body cell would contain 46 chromosomes arranged in 23 pairs. Now, I stress that as a body cell, because if it was a gamete, which we know is a sex cell like a sperm or an egg, that would only have 23 chromosomes, not 23 pairs. And then, of course, one chromosome of every pair is from each parent. So that could be chromosome 1 from your mum. That could be chromosome 1 from your dad. And then the chromosomes themselves are made of tightly coiled strands of DNA. And that a gene is just a segment of DNA, a length of DNA that contains instructions to make proteins, the building blocks of life. So hopefully you're able to do that. So let's move on now. This is another exam question that I've edited here. This is where you're asked to label the diagram. And again, explain what this diagram is showing. Now, this is a very similar question to this one. So how would you label that diagram for 5 marks? What would you give A, B, C, D, and E? How would you explain that diagram for 6 marks? Try and have a go yourself. Pause the video and give yourself ample time to try and answer those 2 questions. Right, okay, so these are your answers. A is a nucleus, B is a chromosome, C is a gene, a length of DNA. So you can see if you were to remove a chromosome, you can see that it's made up of supercoiled DNA and that a length of that DNA is your gene. D are amino acids, and then amino acids join to make a protein. So what is that diagram showing? Well, if you had to put words in a paragraph together to describe that diagram, you could say, well, in eukaryotic cells, you have a nucleus. And within that nucleus, you have chromosomes. If this was a human, you'd have 46 chromosomes, 23 pairs. Chromosomes are made of supercoiled DNA, and that DNA, okay, provides a genetic code. A short length of DNA is called a gene, and a gene provides the instructions to make a protein. That gene determines which amino acids join together to make a protein and how many amino acids join together to make that protein. So it basically determines there are 20 naturally occurring amino acids. It determines which of the 20 join together, the order in which they join, and how many join. And that will give rise to a very specific protein. So this diagram, if you were struggling, you could look back at this slide or even at this one to help you. So moving on. This is a quick checkpoint about genes. So what I would like you to do is I'd like you to read that paragraph, use the missing words at the bottom to see if you can complete that paragraph on genes. Pause your video until you've had a go at it yourself, and then when you're ready, resume your video. Right, okay, so first one, a gene is a small section of DNA found on a chromosome. Each gene codes for or tells the cell what to make a particular sequence of amino acids, and these are joined together to make a specific protein. We've only got 20 amino acids, but they make up thousands of different proteins. I should mention here that not all proteins will contain all 20. It depends on the protein. So DNA also determines which proteins a cell produces. For example, hemoglobin is a protein that binds reversibly with oxygen. It's what carries oxygen around the body. And that in turn determines what type of cell it is. For example, we know that hemoglobin is produced inside a red blood cell. So the genes that are expressed, in other words, the genes which are used to make proteins in a cell will determine what cell or type of cell it is. So if the gene is expressed for hemoglobin and that cell produces hemoglobin, we know therefore it is a red blood cell. So the Human Genome Project, or HGP. Now, what is a genome? What do people mean by the word genome? Well, the genome is basically a fancy term for the entire set of genetic information in an organism. Now, the Human Genome Project was started at the end of the last century, and it had several aims. The first to work out the order or sequence of all the 3 million base pairs in a human genome, so all the A's, T's, C's, and G's. It was to identify all of the genes, and it was to develop faster methods for sequencing DNA. In other words, determining the sequence of the A's, the T's, the C's, and G's for individuals. Now, the sequencing project was finished in 2001. However, work still continues to identify all the genes in the human genome. And the Human Genome Project actually used the DNA of several people to get sort of average sequence. But each person has a unique sequence unless they've got an identical twin. So I don't have an identical twin. So your DNA, your unique sequence will be very different to mine, okay? So mapping of a person's genome can help in predicting how likely they are to have an inherited disease or develop certain conditions like cancer. So you might have heard there was a lot of press coverage about Angelina Jolie and the breast cancer gene. Well, people can actually sequence their DNA and work out how likely they are to develop diseases and cancer, okay? And scientists and doctors are now beginning to use the information they have discovered in the Human Genome Project to make more effective medicines, because the effectiveness of medicines can actually be affected by the different versions of genes that we have. Now, the different versions of a gene are called alleles. If I gave a very simple example of an allele, so humans have different versions of the eye color gene, and the different versions of the eye color gene are called different alleles of the eye color gene. So we have the blue eye color allele, the green eye color allele, the brown eye color allele. Well, it's not just eye color that we have different versions or different alleles for. Lots and lots of genes in our body we have different versions of. And sometimes medicines will work better for someone with one allele than a different type of allele. So if we can sequence the human's DNA, we can find out what alleles they have for specific genes and which medicines will be more effective for them. Now, this is a high question that's appeared in the past. It said all modern human beings are descended from a common ancestor who lived in Africa. How are scientists using genomes to track the migration of certain populations of people around the world? So I want you to think about that and think about what you might put down for a question like that. Pause your video now. Some of you might have seen these advertised where you can actually find out your unique ethnic mix and work out where your ancestors have come from. Well, this is based on the principle that we're going to describe here. So there's lots and lots of different companies that do this, but they literally will sequence your DNA and they will look for certain mutations. And it's those mutations, they are completely harmless mutations that can quite often give rise to different alleles, and sometimes they don't at all. Sometimes mutations are completely silent, that will enable these people to decipher how your ancestors migrated from where they first evolved in Africa. So this is what they do. So we've mentioned the human genome is the entire set of genetic material of an organism. So for a human, the genome is mostly identical in all human beings. That would give you one mark to say that it's mostly identical in all human beings. But as humans actually migrated away from Africa and different populations migrated in different directions, sometimes mutations occurred. And that changed their genome. That changed the order of those A's, T's, C's, and G's. That's what mutations do. Mutations will change the order of the A's, the T's, the C's, and G's. Now, sometimes those mutations are selected for and that they exist within that population. And what scientists do is they investigate those differences, because you can pass those mutations down to your offspring and they pass them on to their offspring, etc. By investigating those mutations, scientists can actually work out when new populations split off in different directions and what route they took. So if we look at this diagram here, if we say that humans, the first ever humans, our ancestors were in Africa here, and the humans migrated in this direction, obviously you're going to have humans migrating everywhere, all over the world. But let's just say that this particular population migrated in this direction. You might have had a mutation occur here, and that this mutation would have been passed on to offspring. Now, that population might have then split. Some might have gone in that direction, north. Some might have gone in this direction here, sort of north east. Some of them would have gone more, you can see sort of southeast in this direction. Those organisms here, and those organisms there would have all carried that mutation A. When they got to that destination, another mutation might have arisen, given mutation B. So those humans there would have been carrying mutation A and mutation B, and you could get that from their genome. Similarly, the population that travelled in this direction, okay, they'd be carrying mutation A, and you might have got another mutation. You might have got mutation E occur. So mutation A and E. And the part of the population that went in that direction, they might have had mutation G arise. Now, what would happen if that organism then, that human being, then travelled over here and settled over here where we are, okay, in the UK, then if you do the genome sequencing, you might see that that organism had mutation G, mutation E, and mutation A. And then you could trace their ancestry and how they migrated. So that's what these companies do. They look for sequences, mutations. And those mutations are characteristic to different populations all over the world. So it's very, very clever how they do it. Right, okay, so what we're going to do now is we're going to do some exam questions to see how much we've learned today. So the first 2 pages are foundation questions. If you want to go straight to the higher questions, then feel free to skip these. But these are grades 3 to 4 questions, they're foundation. So we look at the first one, question 1. DNA makes up the genetic material in animal and plant cells. Which one of the following statements about DNA is correct? So have a read of those and then decide which one that you're going to tick. Question 2, which one of the following statements defines what a chromosome is? And again, just select one. Right, pause your video while you're reading that, and then when you're happy to resume, press play. Right, okay, so the answers to that one are as follows. DNA is found in the nucleus of animal and plant cells, and chromosomes are the structures that contain DNA. So grades 4 to 5, these again are foundation questions, and these are past paper questions. It says question 3. The following diagram shows part of a DNA molecule. Describe the overall structure of a DNA molecule for 2 marks. B, DNA contains lots of sections called genes. Describe the function of genes for 2 marks. Now that is a function, means a job, the role. Question C, what is meant by the term genome? Give one reason why it's important for scientists to understand the human genome. Right, pause your video and then have a go at these questions. And when you're ready, press play. Right, so a DNA molecule is made up of 2 strands of DNA coiled together into a double helix. If you remembered a lot of the information from the slide earlier on, you might have included even more detail. But your main thing there is DNA is a polymer. It's made of 2 strands. We know they're called polynucleotide strands, and they're coiled together in the shape of a double helix. Right, B, DNA contains lots of sections called genes. Describe the function of a gene. Well, a gene is a small section of DNA found on a chromosome, and genes code for particular sequences of amino acids, which are then put together to make specific protein. So describing the function is saying the job or the role it plays. And that's why, okay, it's important to say that they code for particular sequences of amino acids and how these amino acids are put together to make specific proteins, because that is a job or function of a gene. So what is meant by the term genome? Give one reason why it's important for scientists to understand the human genome. Well, there's lots of things you could have put down for why it's important for scientists to understand the human genome. But if you've gone with the one that I've given you in the earlier part of this tutorial, hopefully you would have said the genome is all the genetic material in an organism. It allows scientists to find genes that are linked to different types of diseases, and hence, remember, we can treat them more effectively with medications, etc. So if you were doing a higher tier paper, you would see very, very similar questions. Sometimes you're asked to give more detail for those questions. Sometimes the way that the question is worded would be slightly different. Usually you have to write a little bit more, okay? But you can see they're quite similar, but you can see the depth, particularly for C, is a little bit more. So outline the function of genes, as we said before. Genes code for particular sequences of amino acids, which are put together to make specific proteins. No difference at all there to the foundation question. But you can see B and C are slightly different. So we know that B, what is meant by the term genome, the entire set of genetic material in an organism, and then they want 2 marks for this one. Give one reason why it's important for scientists to have an understanding of the human genome, but explain your answer. Well, on the foundation paper, they ask, give one reason why it's important for scientists to understand the human genome, but they didn't ask you to explain it. Here you do. So you'd still say, okay, it allows scientists to identify genes that are linked to different types of diseases. But this is where you get your second mark. It's for saying knowing which genes are linked to inherited diseases could enable us to develop effective treatments for them. So that's where you get your second mark. So that's your explanation. So this is why it's important, and this is explaining why it's important. Right, so hopefully you've learned a lot from this tutorial. Thank you for listening, and good luck with your examinations.